The same properties that made PCP a bad medicine made it an extraordinary probe of the brain. I really want you to think about that for a second. We're talking about phencyclidine. You probably know it from the streets as angel dust. It's this incredibly infamous drug, universally recognized for its dangers. But hiding right behind its terrifying cultural reputation is honestly the ultimate paradox. This notorious substance, which started out as a completely failed surgical anesthetic, secretly became psychiatry's absolute most important scientific instrument.
Welcome to this explainer. Today, we're going to cut through the street mythology and look straight at the hard science. We are taking a deep dive into the hidden medical legacy of phencyclidine to see exactly how it reshaped our entire understanding of the human brain. And trust me, the neuroscience we're about to get into is just absolutely fascinating. All right, let's just dive right into this. Here's our roadmap. We'll cover its origins as a failed anesthetic, how it uniquely plugs the NMDA-receptor, its role as a schizophrenia simulator, the chaos of cortical-disinhibition, its real clinical harms, and finally, how it paved the way for ketamine.
Starting with Section 1, the failed anesthetic. To really understand how PCP changed neuroscience, we first have to understand how spectacularly it failed in the operating room.
Okay, so picture this. It's 1956, and PCP actually begins its life with incredible promise. Synthesized over at Park Davis, it was introduced as an IV anesthetic called Sernial. And it honestly seemed like a massive breakthrough, mainly because it didn't depress a patient's breathing or heart rate like the older drugs did. But, yeah, by 1959, researchers were already noticing some bizarre schizomimetic, or basically psychosis mimicking effects. When used in surgery, it was a disaster. Up to a fifth of patients woke up delirious, agitated, and hallucinating, sometimes for hours on end. Sernial had to be completely abandoned. But instead of just giving up, chemists went back to the lab and developed a shorter-acting, safer alternative in 1962. You've probably heard of it, ketamine. But the scientific story of PCP didn't end there. It was actually just waiting for a massive discovery to drop in 1983.
Which brings us to Section 2, plugging the NMDA-receptor. This laboratory breakthrough from 1983 completely revolutionized how we view this entire family of drugs.
The absolute defining discovery here was that PCP and ketamine are what we call NMDA-receptor antagonists. But they don't just work in a standard way. They use a highly specific mechanism called open-channel blockade. So imagine an open door in your brain's circuitry that allows ions to flow through. PCP binds to a site deep inside that open pore and acts like a literal plug in the doorway. Because it physically obstructs the channel, it can actually only reach its target when the receptor is actively open. This means the drug literally punishes your busiest neuroreceptors. The more active the brain circuit, the more heavily it gets blocked.
Now, if we rank these blockers by their potency, a research chemical called MK801 is the absolute strongest, right? That's followed by PCP and finally ketamine. So PCP is a far heavier, much longer-acting blocker than ketamine, but it doesn't even stop there. It also acts on secondary targets. It inhibits dopamine transporters and engages sigma and nicotinic receptors. And these secondary effects, they are exactly what give the PCP experience its intense, euphoric, and deeply psychotogenic coloring. Let's look at Section 3, the schizophrenia simulator. Because this is exactly where PCP's story shifts from, basic pharmacology into a really profound psychiatric revelation.
And this brilliantly illustrates why PCP became a sort of master key for understanding severe mental illness. See, for years, psychiatrists would use amphetamines to model psychosis in the lab, but amphetamines really only mimic the positive symptoms of schizophrenia, meaning the added behaviors like paranoia or hallucinations. The PCP and ketamine model does something practically unmatched. It reproduces those positive symptoms, sure, but it also reproduces the negative symptoms, like emotional blunting and social withdrawal, and the cognitive deficits, all at the exact same time. It quite literally recreates the entire clinical picture of the illness in totally healthy volunteers.
So why does this matter so much? Well, this single observation launched what's known as the NMDA hypofunction hypothesis. Researchers basically realized, hey, if giving someone a drug that blocks NMDA receptors creates a flawless simulation of schizophrenia, maybe endogenous underactivity, like a natural lack of NMDA signaling in the brain, is a root cause of the illness itself. This idea completely reframed the field's thinking. For the first time, it gave neuroscience a real way to explain those negative and cognitive symptoms that the older dopamine-only theories simply couldn't answer.
Moving to Section 4, cortical disinhibition and chaos.
Now, stick with me here, because to really grasp how this works, we need to look under the hood at one of the most elegant ideas in modern circuit neuroscience.
Let's ask a pretty fundamental question. If NMDA is an excitatory receptor, why does blocking it cause a state of hyperactive, chaotic overexcitation in the brain? It seems like a biological paradox, doesn't it?
Well, the answer lies in exactly which cells get blocked first. Think of it like the brakes on a car. First, PCP specifically targets highly sensitive NMDA receptors that are located on parvalbumin-GABA interneurons. These are essentially the brain's brake pads. So, by blocking these receptors, the drug essentially cuts the brakes. Without that restraint, the main excitatory cells, the pyramidal neurons, are just completely released. They start firing totally unchecked, causing this massive paradoxical surge of glutamate. It leads to incredibly disorganized, poorly synchronized cortical activity. So the sheer felt chaos of a PCP experience and the persistent architecture of clinical psychosis, they actually share this common root. The wrong cells went quiet.
On to Section 5, Pharmacokinetics and Real Harms. Because obviously we can't talk about PCP without addressing its real-world dangers using some sobering, unsensational clinical data.
So, you've definitely heard the street myths about angel dust, right? How it gives people superhuman strength or causes these unstoppable violent rampages. But let's look at the absolute best data set we have, a landmark series of 1,000 consecutive PCP intoxications. Out of those 1,000, do you know how many actually featured violence? 35%. Now, don't get me wrong, that is a very real and significant number, but it completely debunks that myth of the universal superhuman rampage. In reality, a lot of patients presented as just calm, wildly disoriented, or even catatonic.
But the actual medical emergencies, they are severe. PCP has this long, highly variable half-life of around 21 hours. And because it stores in your fat tissue, people can experience reintoxication days later as the drug just slowly seeps back into their bloodstream. The real physical dangers include rhabdomyosis, which is this brutal, severe muscle breakdown that can completely destroy the kidneys, and life-threatening hyperthermia, meaning extreme overheating. This is exactly why modern emergency treatment heavily relies on a calm environment and chemical sedation with benzodiazepines. Putting physical restraints on a highly agitated, hyperthermic patient, that actually just worsens the muscle breakdown and the heat injury.
Which brings us to our final part, Section 6, paving the way to ketamine. This is where the dark history of a super dangerous drug finally pays off.
So, the absolute crucial takeaway here is this. PCP itself has absolutely zero therapeutic use today. None. It is way too long-acting, it's way too toxic, and it's far too prone to causing psychosis. But intellectually, it was the ultimate scientific Trojan horse. The discovery of the NMDA receptor's role, which we only figured out through PCP, directly led researchers to ask a wild question. Could manipulating that exact same receptor treat severe mood disorders? That one insight seeded the glutamate hypothesis of depression. And the result of that? The 2019 FDA approval of S-ketamine for treatment-resistant depression. Basically, PCP was the drug that found the door, and ketamine was the drug that was safe enough to actually walk through it.
Which leaves us with this final, kind of provocative thought. If a completely failed, wildly hazardous 1950s anesthetic could inadvertently map out the circuitry of schizophrenia and lay the groundwork for the most significant antidepressant breakthrough in decades, what other failed drugs are just sitting on laboratory shelves right now, quietly hiding our next great medical breakthroughs? Thank you so much for joining me on this explainer. I really hope this changes how you view the messy, unpredictable, and just incredible nature of scientific discovery.